evening, class. How is everyone doing?
Yes, my tired, miss.
Doing well, miss. And how are you doing?
I'm doing well. Sabrina, how are you?
Tired, but I'll be paying attention, miss. So go ahead.
I appreciate it as always. So class, today our lesson is going to be on recombinant DNA technology. To start, would anyone like to tell me what they think recombinant DNA technology is? Elon, what is do you know what recombinant DNA technology is?
No, miss, what is that?
Kevin,
Using scientific methods, skills, equipment to recombine DNA to make a new species or a new DNA structure.
That is a very good attempt.
Sabrina, would you like to try?
I agree with Kevin.
Okay. So we have some specific objectives for our lesson today. I hope we can get through the majority of it. The first would be to outline the principles of restriction enzyme technology, or in removing sections of the genome, explaining the steps in recombinant DNA technology, and if we have the time, to discuss the importance of genetic engineering. So we jumped ahead a little bit with talking about recombinant DNA technology, but Kevin, your definition was very good. So we'll get back to that and dissect that definition a little bit more later. So to start, we're going to look at what is genetic engineering. To have a good understanding of recombinant DNA technology, it's a type of genetic engineering. And so in genetic engineering, we're changing the DNA characteristics of an organism. And we do this by modifying genetic material. And when we change the genetic material or the DNA characteristics, this will produce an outward change in the organism, because our genetics corresponds to a phenotypic trait. Is everyone following so far? I know I taught you guys this at CXC.
Yes, miss.
So the video is running a little bit ahead of me. But yes, recombinant DNA technology, as I said, it's a form or a method of genetic engineering where we're recombining different pieces of DNA in order to produce desired characteristics in an organism. So I think after watching this brief video, you guys will understand better.
Recombinant DNA technology is a technique in which a fragment of DNA from a donor cell or organism is isolated and then inserted into the DNA of another cell or organism. This allows scientists to introduce a new characteristic into an organism by inserting a new gene into it. Recombinant DNA technology involves 4 major steps. Firstly, a DNA fragment containing the gene of interest is obtained from a donor cell. Secondly, a suitable plasmid is obtained from a bacterium. Plasmids are commonly used as vectors to transfer the gene of interest into a host cell for expression. Thirdly, the DNA fragment containing the gene of interest is cut using an enzyme called restriction enzyme. This enzyme acts like scissors. It recognizes a specific base sequence and cuts the DNA at a specific point. On the other hand, the same restriction enzyme is used to cut open the plasmid. Lastly, the DNA fragment containing the gene of interest is inserted into the open plasmid with the help of another enzyme called DNA ligase. This enzyme acts like glue. It catalyzes the joining of the DNA fragment and the plasmid. A recombinant plasmid is finally formed. This recombinant plasmid can then be introduced into a host cell for different purposes, such as producing proteins of other species like human insulin, or producing genetically modified organisms that possess new characteristics.
Okay, so after the video, does everyone understand what recombinant DNA technology is a bit better?
Yes, miss.
Yes, miss.
Ellen, I don't believe you're sleeping in my class. Young man, wake up, please. So, Kayla, would you like to tell us what a genome is? Okay, so it's just a simple one to you're following along with the screen.
Okay. All the genetic information of an organism.
Okay, good. That's a genome. Kevin, would you like to read what a plasmid is? Kevin, are you with
Yes.
us? You like to
Yes,
read what plasmid is?
Okay, a plasmid is a genetic structure in a cell that can replicate independently of chromosomes.
Okay. Sabrina, would you like to read what gene therapy is?
Is by correcting the underlying genetic problem.
Ellen, would you like to read what restriction enzyme is?
Enzyme produced chiefly by certain bacteria having the property of cleaning DNA molecules at or near a specific sequence of bases.
Okay, good. So with having these definitions, we can better understand recombinant DNA technology. So the change that is happening in recombinant DNA technology is a change that is affecting the genome, which is where all the genetic information for an organism is stored, right? So as seen in the video, the first thing we're going to do is we're going to use restriction enzymes, which are enzymes produced by bacteria that are able to cleave DNA of molecules. This is usually something the bacteria use to protect themselves against other predatory bacteria or viruses. So this technology has been exploited in recombinant DNA technology. So we'd use a restriction enzyme or enzymes to cut a DNA, a specific sequence of DNA. Then there's a structure called a plasmid, right? This is a genetic structure which it can replicate independently of chromosomes. So you'll find plasmids in bacteria as well, but they are able to replicate independently of the rest of the bacteria. So while the bacteria is doing one thing, the plasmid can do its own thing. We're going to use the same restriction enzyme and we're going to cut the same size piece away, remove that from the restriction enzyme, and that piece of the genome from another organism that we cut, we're going to put it in place of that missing piece from the plasmid. And we're going to use another enzyme called DNA ligase to join the two together so that this compound, or not compound, but this structure that we've created is now going to be stable. Then we can put this plasmid into another organism, such as another bacteria. And at the rate at which bacteria are able to reproduce, the bacteria are going to recognize this plasmid, they're going to read the genetic material, and they're going to produce likely a protein. And this protein will be something that is of extreme importance to us, such as human insulin. The bacteria are able to produce a lot of this protein. And we can use that for various applications, such as in medicine for producing human insulin. You know, that's very useful for diabetics. It's frankly life-saving for them. Or in gene therapy, which is a medical approach where we treat genes by trying to correct the underlying genetic problem. Most of the research using gene therapy and recombinant DNA technology is still in its, I want to say, an early enough stage, but there are a lot of advancements. This might be something you guys would consider as a career. A lot of money there. So with this technology, there's so many different things that we can do. One of them or another of them would be even engineering plants. So Kevin, let's say you're partnering with your grandfather and your grandfather grows OTT apple. We know the skin on OTT apple is very, very soft. You touch it 2 times and it bruise, right?
Yes, miss.
Yes. So let's say we want to make the skin thicker like American apples, right? So what we're going to do, we want to insert a gene from, let's say, a Granny Smith apple. That's the green American apple. Let's say we're inserting the gene that would correspond to producing a thicker skin, right? What we're going to do is we want to introduce that gene into the genome, that specific gene sequence into the genome of the OTT apple, right? We do that, we grow a few trees, or maybe we start with tissue culture and then put the trees in the field and they grow up. And we see these apples, they're nice, they're big, they're red, they're juicy, and they have the thick skin. You know how that benefits us? We can export these apples now. We can earn a lot more money because by the time them reach your auntie and uncle in foreign, all the Jamaicans that miss home, they're not bruised up or mashed up or spoiled because they have this thicker skin now and they're able to survive that long transport. So you see how that's able to help you?
Yes, miss.
Okay. In your own words, would you like to go over the steps of recombinant DNA technology?
Obtaining a plasmid, right? Obtaining the desired gene sequence that you like. Integrating that gene sequence into the plasmid using restriction enzymes and DNA ligase, and then introducing that plasmid into a desirable host such as an E. coli cell, right? Allowing that plasmid to, let's say, increase in numbers in that cell, and then introducing that plasmid to a introduce it to a species, whether it is the apples that you spoke about or the carrots or anything, for the genes to be expressed, thus giving the species or the fruit new features, desirable features that you want.
That is an excellent explanation. I'm very proud of you. Okay. So, as said before, we'll have a little bit of time to further go into the applications of this technology of genetic engineering. Would anyone like to state a possible downside to genetic engineering that they can think of? Don't all answer at once.
A possible downside that to do with the ethics behind of it. So it could be a case that you create a new species of plants, right? And that species of plant, let's say breeze blow, the pollen grains blow somewhere, or the seeds are dispersed, and they end up in nature, and they end up being quite combative to the environment and competitive to other species, thus giving way to their rise and the decrease in population of plants that were here a long time. So you would end up having a shift in the ecosystem.
Yes, that is a great example of something that can be the negative side of genetic engineering. Have you guys begun to appreciate the importance of genetic engineering? Dr. Kayla?
Yes, yes.
Yes, miss.
Good. So does anyone have any questions before we close?
No, miss.
Okay. So for homework, we have a quizzes exercise. It's just a short quiz with a few questions on recombinant DNA technology and genetic engineering. Should be nothing too hard for you, Bright Sparks. So please copy the link before you leave the session. And at the start of next class, Ellen, who has been sleeping out the class today, will be explaining the topic for everyone.
Okay, miss.
Thank you for
Thank you, miss.
listening.
All right.